One, thank you for being here on time. I appreciate it as always. Morning, Anna. Hello, the folks in 10 Evans. We have a guest speaker today, as you know, so this opening few remarks shall be very brief. I asked Maxine and Andre and Katarina who these are, and it's not surprising that most folks don't know who these are, but you certainly know what they did. So that fellow here is the English poet John Keats. He wrote Ode to a Nightingale. That's Emily Bronte. She wrote Wuthering Heights. That's the composer Frédéric Chopin. And that's the writer Anton Chekhov. Chekhov is not the character on Star Trek, the other Chekhov. I don't suppose any of you know what these numbers mean. The age of their deaths. I don't suppose any of you know. So Keats died at 25. Emily Bronte at 30, Chopin at 39, Chekhov at 44. Anybody know what they died of? Tuberculosis. Good for you. They died of TB. Now, the 19th century saw an astonishing plague which took away tens of millions of people. Plague. But it also saw tuberculosis just sweep away entire generations, not just of incredibly brilliant and famous people, but people. As we fast forward to 2012, there is both good news and bad news. First, the good news. The good news is, according to the CDC, in the United States of America, 547 people died of TB in 2009. Now, every single death, of course, is a tragedy. Having said that, it is pretty clear that the mortality rate from tuberculosis in the United States today is a microscopic fraction of what it was in the 19th or really even early in the 20th century. Now, this doesn't mean that TB is no longer a problem. It's a huge problem. 1.7 million people died of it. But their deaths have not to do with our inability to treat the disease. It's our, scientifically or medically, their deaths have to do with our inability as a group of people, as humanity, to get the drugs and the treatments to the people in most need. Because the treatment, of course, exists. Some of you probably haven't had a chance to study the ribosome, and too bad. Not only will the ribosome be on the midterm, I promised you it would be on life. And it is, in fact, the case that Selman Waxman, shown here, hello, paging pointer appears. Selman Waxman and his student Albert Schatz, yet another of the many examples in which a famous scientist and a student collaborated on an effort, together purified this compound, and Waxman won the Nobel Prize for that. The compound is streptomycin. It was the first antibiotic shown to be effective against TB, and this paper, if you pull it down, which I think you should, and look at it, will tell you that, in fact, streptomycin kills the bacterium that causes tuberculosis by inhibiting its ability to translate proteins. Ask yourself the question, then ask yourself that question at office hours or ask your GSI. Why doesn't streptomycin inhibit our protein synthesis? So we take a person with tuberculosis, give them streptomycin, and the bacterium dies because it can no longer synthesize protein, but the human being doesn't. Why is that the case? How can you inhibit a general process, such as translation, and yet not have overt toxicity on the host? How can you have such selective action? Tuberculosis, at least in the United States of America in 2012, is, putting it mildly, not a major source of mortality. And I realize it's kind of early to talk about death, but we have to be realists. What do people die of today? Well, the latest comprehensive statistics available are for 2009. 2.43 million people died in this country in that year. 600,000 approximately died of heart disease. 137,000 of chronic lower respiratory diseases. 130,000 got strokes. 120,000 died in accidents. Alzheimer's took away 80,000 people. And you will notice I skipped the entry in red. The C, cancer. Cancer took away 570,000 people in this country. And the challenge is, therefore, for your generation of physicians, nurses, pharmacists, scientists, physician scientists, public health professionals, to do to cancer what the physicians and scientists of the 20th century did to tuberculosis in the 20th century. That is, dramatically lower its incidence and mortality rate. So you may think that the quiz you're about to take has to do with the lac operator. In fact, it doesn't. As today's guest lecture will convince you, this is teaching you in preparation for that rather majestic task. But before you can dive into that rather majestic task, we must take the quiz. And the quiz is, as always, one of these is wrongity wrong wrong. A, and of course, as promised, this is about the lac operon. The genes LAC-Z, LAC-Y, and LAC-A are transcribed as one messenger RNA that is then translated into 3 polypeptides. This ensures that comparable amounts of the 3 enzymes are made. B, the promoter defines where RNA polymerase starts transcription, but not whether it will do so. That is done by the joint action of the operator and the repressor. C, the operator is a DNA sequence that is bound by a protein, the LAC repressor, encoded for by the LAC-I gene. D, the transcription and translation of the LAC-I gene are controlled by levels of the inducer, lactose, in the growth medium of the bacterium. And E, when the LAC repressor is bound to the operator, RNA polymerase cannot transcribe the, sorry, not LAC-X, I beg your pardon, that's not the, not the, no, LAC-Z, sorry, the LAC-ZYA genes into messenger RNA, okay? All right, let me get going on this. As always, I'll give you 1.5 minutes. All right, folks, and without further ado, as always, ask your neighbor, Google, PubMed. All right, 45 seconds down, 45 to go. Another 15 seconds and then be done with it. All right. 5, 4, 3, 2.5, 2, 1.5, 1.5, 0. Good job. Let's see. Very nice. Good for you. As your book describes carefully, the LAC repressor is always in the cell. Transcription and translation are not the controlling element here. The way the circuit works is the small molecule, the inducer, controls whether the repressor is active or not. The control is entirely post-transcriptional and post-translational. Good for you. Well done. Okay. Let me quit out of this, quit out of that. And I only have 2 more slides. Please, thank you, please go away. And without further ado, we can transition to we are powered by small molecules that control gene expression. Many of you asked me, what are these small molecules that have to do with transcription and diabetes? There you go, Avandia. It's a ligand for PPAR. Go to PubMed, type PPAR and diabetes, and you'll see what I'm talking about. This is a very potent anti-inflammatory drug, dexamethasone, skin rashes, asthma, that sort of thing. This is chemically a very different creature, but it's essential for the development of cognition, for example, in humans. It's thyroid hormone. So today's guest speaker, I'm delighted to say, has had a remarkable career pushing as far ahead as anyone ever thought possible our frontiers of understanding of how small molecules and other things, as you shall hear shortly, converge on the development of disease, specifically cancer. So Dr. Mond, whom I'm delighted to introduce, is a native of the Bay Area, got her undergraduate degree at Pomona and her PhD at Wake Forest, and is at the Stanford University School of Medicine, doing to cancer what the 21st century has to do, or else a century from now, we shall still be looking at the same grim statistics. The reason I asked Dr. Mond to speak to you today is because I want to frankly inspire you and show you that this can be done. This is real. There is a world outside the ivory walls of academia, and in that world, people are doing remarkably next-generational things to push forward our translational efforts in eliminating the public health scourge of cancer. Without further ado, it's a pleasure to welcome Dr. Mond to Bio1A. All right. We need a minute to get set up for me. Your way. All right.
Good morning. All right. Thank you, Dr. Ernov, for that introduction. I'm honored to share the stage with you here, and very excited to be here. This was not on my CV that I gave you, Dr. Ernov, but did want to mention that even though I do work at Stanford, I have an abiding devotion to Cal. I went to preschool here while my parents were graduate students and faculty, and so yeah, don't throw things at me. All right, so cancer. Cancer is, as some of you may know, the Latin word for crab, which is why it's also the name of that constellation. And doctors in Greece back in the 2nd century, when they opened up a tissue and saw a tumor in it, they saw this mass that had these extensions into the surrounding tissue environment that looked like crab legs sticking out. So I don't know if you can see that resemblance there. Anyway, that's why they call it cancer. So this is a prostate. This is what I work with in the lab. We get about 2 to 3 prostates from radical prostatectomies per week. Apologies to those of you still eating breakfast, but if you're going to be going into any health-related fields, medicine especially, you will definitely get used to seeing gross things while you're eating. You can see this prostate here. You can actually see some of the cancer right here in the middle of it, which is, yeah, pretty nasty. But here's a less bloody picture of a prostate, and, well, an illustration, that is. And the prostate gland is located below the bladder, and it envelops the urethra and the seminal vesicles. And it functions to synthesize proteins and secrete proteins into the seminal fluid that helps protect the sperm and facilitate insemination and fertilization. Now, of all of the cancer, or types of cancer, prostate cancer is one of the most frequently occurring cancer in U.S. males. And it's also the 2nd leading cause of cancer deaths in U.S. males. These are the major risk factors for prostate cancer. Age is the biggest risk factor for prostate cancer. 1 in 6 men will get prostate cancer in his lifetime, and that risk increases with age. Race is also a factor. African American men have among the highest risks for prostate cancer of all populations, and their mortality rates from prostate cancer are over twice as high as those in Caucasian men. Geographic location also comes into play. Men who live in Asia have among the lowest prostate cancer risks in the world, And interestingly, men who live in Asia but then move to the US then have an elevated risk for prostate cancer. And of course, this is likely due to environmental factors such as the American diet versus the traditional Asian diet. And then lastly, family history also comes into play, as it does in many cancer types. And men who have had family members with prostate cancer can have up to a 45% higher risk for developing prostate cancer themselves. And so among all of these risk factors, I did want to point out that both genetic and environmental factors can contribute to prostate cancer risk as well as its severity. Briefly, here's some of the common treatments for prostate cancer. If the cancer is detected at an early stage, as in if it has not left the prostate, or it's confined to the prostate, then it can be removed through surgery or shrunk through radiation, and there's essentially a 100% 5-year survival rate, which is great. However, if the cancer is detected at more advanced stages or has metastasized, then the survival rate, the 5-year survival rate decreases to about 45%. And there's no, the cure, or there's no real cure for this advanced form of prostate cancer, and patients can undergo hormone therapy, radiation, chemotherapy, most recently immunotherapy, or a combination of those. So back to the prostate biology. The prostate is a glandular organ that's composed of a bunch of branching ductal structures, similar to the mammary gland. And each of these prostatic ducts, here's sort of an illustration of a cross-section of a prostatic duct. It's composed of a number of different specialized prostate epithelial and stromal cells. So here we have a layer of luminal prostatic epithelial cells, which are functionally differentiated to be the cells that secrete proteins into the luminal space. Then you've got a layer of basal epithelial cells that separate the epithelial compartment from the stromal compartment. And then the stroma is it's the microenvironment of the prostate, and it consists of a number of cell types. You've got the fibroblasts, blood vessels, collagens, et cetera. And I did want to point out that the cells in the adult prostate, the fully developed adult prostate, are differentiated, and they are non-dividing. So in terms of the cell cycle, they are in G0. Okay, they're not actively dividing. Now, during prostate cancer progression, these differentiated specialized prostate cells undergo what's called dedifferentiation, where they lose that functional specificity, and this contributes to cancer progression. So one of the early stages in cancer progression in the prostate is characterized by hyperproliferation of the luminal epithelial cells into the luminal space of the prostatic ducts. And this is called benign neoplasia, and it is still a benign condition because these cells have not breached the prostatic duct structure. Now, once these cells hyperproliferate and invade through the prostatic duct into the surrounding stroma, then that is characterized as cancer and is malignant. And then as this further progresses and gets more advanced, then you get a complete lack of any structural differentiation of the original prostate. You know, there's no more of these functioning glandular structures. It's just a big mess of cells and infiltration from the stroma and inflammation and all sorts of cancerous components, which just further propagate the aggressiveness of the disease. And so 2 of the major questions in cancer research, and that we'll be addressing in the lecture, is 1, what causes this dedifferentiation or transformation to happen? And 2, how can we stop or prevent it? So genes play a big role in development and progression of cancer, and more specifically, the mutations of genes. And so these mutations can, or for the most part, can occur in a sporadic manner. And by definition, this means that sporadic mutations do not appear to be inherited mutations. They can appear at random to any cell in the body. And these mutations are caused by random alterations to genes that end up affecting their expression levels one way or the other. Some examples of how this can happen are the exposure to environmental carcinogens such as UV rays or cigarette smoke that can cause DNA damage, which can disrupt normal gene expression. Mistakes in DNA replication can result in mutations that also affect gene expression. Rearrangement of chromosomes can interrupt various genes and again cause mutations in them. And also introduction of viral genes into the host DNA that can also interrupt genes within the host DNA and cause mutations that can cause cancer. On the other hand, you also have a class of mutations called familial mutations, in which these are mutations that are heritable. They run in families. And individuals who have these mutations have an increased incidence level for developing cancer due to that inherited mutation. However, just because an individual has inherited a mutation does not mean that they will necessarily get cancer. There are different levels of penetrance for these types of mutations, and you may still be able to prevent cancer through healthful lifestyle or by just having good compensation mechanisms for that gene mutation. So your book illustrates this concept in this figure here, and they're showing an example of sporadic and familial mutations with the gene BRCA1, which I believe Dr. Ernov has spoken to you about already. So an example for sporadic mutations of BRCA1, the individual is born with 2 normal copies of the BRCA1 gene, and if one of them is mutated, you still have a normal phenotype. You get normal cell growth. And it's not until the second copy of the gene is mutated that it can then help cancer progression. Now, this is in contrast to those with an inherited mutation of BRCA1. These individuals are born with already having one mutated allele of BRCA1, and it only takes one more mutation to mutate the other allele, which then leads to cancer progression, or can at least aid in cancer progression. And BRCA1 it's probably one of the more well-known genes involved in cancer progression, and sometimes the media refers to it as a cancer gene. But that's sort of a misnomer. If anything, it should really be called an anti-cancer gene or a tumor suppressor gene. And that's because the normal function of this gene is to keep cell growth in check. It's to help regulate the progression through the cell cycle and make sure that it doesn't get out of control. And these genes, when they're mutated or inactivated, then they lose that control, and that makes cells more susceptible to uncontrolled growth. Does that make sense? Another example of a common tumor suppressor gene is P53. P53 is mutated or inactivated in about 50% of all cancers. And P53's normal functions, again, are to help induce cell cycle arrest at G1 and G2 of the cell cycle. And this is particularly important, for example, if from UV rays you get some DNA damage and the cells want to stop dividing in order to repair that DNA damage before continuing on to replicate the cell. And P53 also aids in DNA repair functions. And if that DNA cannot be repaired, rather than continuing on in the cell cycle, P53 helps signal those cells to die or undergo apoptosis. And so you can imagine that this is a pretty important function for one gene to have, and if that gene is mutated, then that can be a problem down the line and, of course, lead to cancer. And I did want to point out that the inactivation of one allele of a tumor suppressor gene is not sufficient to get a cellular effect. You actually need to have inactivation of both alleles in order for the tumor suppressor effect to be abrogated. And some examples of how tumor suppressors can lose their effect are through loss of function mutations, through transcriptional inactivation of these genes, as well as just deletion of chunks of chromosomes that have these tumor suppressor genes encoded in them. Another big class of cancer-related genes are the proto-oncogenes. And these genes normally function to help cells progress through the cell cycle and help them survive. And if these genes are mutated, or in which case mutated in terms of if they are overexpressed or amplified or activated when they should not be activated, then that's when they are called oncogenes, and they contribute to cancer initiation and progression. And in this case, alteration of just one allele of a proto-oncogene is sufficient for cellular effect. And so an example of a common oncogene is MYC. And MYC normally functions to help cell cycle progress through the G1 to S phase. And it also helps to inhibit cell death, inhibit apoptosis. Now, oncogenes can be amplified or activated in a number of ways. For example, again, chromosomal translocation can cause overactivity of a gene when it usually is not active. Gene amplification can also cause elevated levels or activity of that gene. Increased transcription of a gene, also same effect. And also there can be constitutively active mutants of proto-oncogenes. And constitutively active just means that they are always turned on. Okay, they're not regulated or turned on and off in a normal way. They're just always on. So here's an example of a gene fusion that is common in prostate cancer that contributes to oncogenic properties of, well, this fusion gene has oncogenic properties that contribute to prostate cancer. And it's a fusion between the first exon of the gene TMPRSS and a truncated form of the gene ERG, and it's called, of course, TMPRSS2 ERG. And this gene fusion product acts as a transcription factor, and it leads to transcription of genes that cause MYC activation, another oncogene, dedifferentiation of prostate cells, and promotes cellular invasion of prostate cells, which can all help contribute to cancer progression. I also want to point out that TMPRSS2 ERG is regulated by hormones. And this is because the TMPRSS2 gene contains an androgen response element in its 5' untranslated region, which means that, and because that region of the TMPRSS gene is fused to the ERG gene, that means that that androgen receptors, once they're activated by binding from an androgen, can recognize the promoter region of this fusion gene and induce its transcription and then expression of the oncogenic gene product. And this is just one example of how hormones can regulate a gene that contributes to prostate tumorigenesis. And hormones are really quite involved in both normal prostate function and prostate cancer development. Androgens, specifically testosterone, are required for normal prostate differentiation, function, and survival. And this is due to, again, its transcriptional activity. So testosterone, when it enters the cell, it's converted to dihydrotestosterone, which is the form that can bind the androgen receptor. This can then enter the nucleus and bind to the regions of DNA that contain the androgen response elements, which are upstream of androgen target genes. And so it can then cause transcription of the genes that carry out the normal functions of differentiation and survival of prostate cells. However, androgens also drive prostate cancer growth. And so one of the common therapies for men with advanced prostate cancer is called hormone ablation therapy, in which they remove the source of androgens via castration. And either physical castration or chemical castration. Most men opt for chemical castration, which means taking drugs that that inhibit the source of the hormones. So now this can be quite effective. Or hormone ablation therapy does cause tumors to regress and shrinks metastases, and that is good. However, it's extremely common for men to get a recurrence of a form of prostate cancer called castration-resistant prostate cancer, which is much more aggressive and unfortunately is as yet incurable. And one of the hypotheses for why this happens is that after hormone ablation therapy, you get rid of all the cells that are responsive to androgens. They die, and they're gone. But it's thought that there remains a small population of prostate cells that are mutants, and that they don't require androgens to grow and proliferate. And this small population of castration-resistant cells can then, over time, slowly grow up to reconstitute a tumor, and again, a more aggressive tumor. Now, one of the most well-described gene targets of androgen receptor signaling in the prostate is the gene called prostate-specific antigen. Many of you have probably heard of this in terms of the PSA test, which maybe your dads have started getting. And PSA is normally present in the blood at low levels, about 0 to 4 nanograms per mil, and it is then elevated in men with prostate cancer. Therefore, it is a very commonly used biomarker for prostate cancer, and that's why men start getting PSA tests after the age of 50. However, PSA is also elevated in men with conditions other than cancer, such as benign prostatic hyperplasia, which is just hyperproliferation of prostate cells that are not tumorigenic. Also prostatitis. For example, obese men tend to have higher levels of PSA in their blood, so that can be confounding. Also, PSA levels do not distinguish between aggressive cancers and non-aggressive cancers. And this can often lead to unnecessary treatment in men who have prostate cancer that may not actually progress to a level that even requires clinical intervention. And I also wanted to point out that some men that have prostate cancer don't even have increased PSA levels, so that would be a false negative on PSA tests. So with that in mind, should men continue to be screened for prostate cancer using the PSA test? Well, it doesn't really sound like a very good test, given all these confounding factors in PSA levels and how it's not really a very good biomarker for prostate cancer, right? But on the other hand, it's the best biomarker we have so far, and PSA testing has undoubtedly saved the lives of many, many men. And sure, it's also caused overtreatment of other men, but at this point, it's really it's a personal decision, and that a man has to discuss with his doctor, and it's really all we can say about it right now. And what we can do about it, though, is try to find better biomarkers for prostate cancer, particularly biomarkers that can distinguish between a less aggressive tumor and a more aggressive tumor. So that is a huge aim in prostate cancer research. Okay, so I've been mentioning how all of these genes can be upregulated or downregulated or turned on or turned off in ways that can contribute to cancer. Well, I did want to point out that it's very rare for just one genetic mutation to cause cancer, okay? So there's something called the multi-hit hypothesis, which means that, well, you need multi-hits or several different mutations over the course of time in order to lead to cancer initiation and progression. And so this is an example from your book in colon cancer, in which the first mutation, for example, the loss of the APC tumor suppressor gene, predisposes the cell to transformation. And then subsequent mutations, such as activation of an oncogene or loss of another tumor suppressor gene, then leads to progression through, maybe through a small growth to a larger growth, and then to a malignant growth and metastases. So with all of these different stages of cancer progression and all of the genetic regulation that goes on to drive each of these stages, it's maybe giving you a little bit of a glimpse into the complexity behind what leads to cancer. And so, why haven't we cured cancer yet? Well, it's because of this complexity, in large part. The more we learn about one genetic, one mode of genetic regulation or one signaling pathway that can lead to cancer, the more there is to know about, oh, well then how is that one regulated? Oh, well then what regulates that? And it's just a bit overwhelming. And one of the reasons, or one of the aims in cancer research to try to better understand what's going on in the human tumor is that we need better models for cancer research. And so most cancer researchers use models such as cell lines, cancer cell lines, mouse models of cancer. Those are the two main forms of cancer models. And while these have undoubtedly given us just a huge amount of information, it's been extremely, extremely valuable. We've come so far with these models. They are still less than realistic. Okay, so cancer cell lines, these are just cells that we grow in dishes in the lab over decades. These cells are not really the cell, they're not actually similar to the cells that are actually in the human tumor. They've kind of, there's a big disconnect there. And so in both cellular and mouse models, there is a lack of cellular and molecular heterogeneity that is present in native human tumors. And furthermore, these cell lines in these mouse models, they lack the natural microenvironment of the tumor that contributes a lot of important signaling messages that help propagate cancer progression. And also, importantly, I wanted to point out that the genetic profile of one part of a tumor may be different from the genetic profile of another part of a tumor, or from a metastasis. And this was kind of addressed in this article I believe Dr. Ernov showed you, in that when you take a biopsy of a tumor, that only gives you a very narrow view of the molecular and cellular signature of that tumor. And so these problems, these are problems because this kind of thing leads to poor drug development, or the drugs, once you deliver them to an actual human, they may not penetrate the tumor or be delivered to cells in the most efficient manner than they could. And furthermore, these drugs, once they're in an actual human tumor, they may act differently than they would in a cell line or in a mouse. And so that's due to altered differences in drug metabolism. And also, these models, we don't get as good biomarkers for prognosis and treatment of tumors than we would from an actual human sample. So to address some of these issues, I'm going to talk to you a bit about the research that my lab is working on, looking into more promising models for studying prostate cancer. And these are based on the concept of organotypic culture. And organotypic culture essentially means that you take a chunk of an organ and keep it alive outside of the human, all right? Either in a cell culture dish, which is in vitro, or put it back into a mouse in vivo. And the in vivo version is called a tumor graft model. And we believe that taking a chunk of the actual tumor and studying that outside of the human is a more accurate way to look at what is going on in the human tumor, because this actually maintains the native genetic and cellular heterogeneity of the tissue in which to test drugs, for example. And our lab has published on the development of the tumor graft model, where you're welcome to look up that paper there. You can go back to the slide later if you didn't get to write that down. I did want to mention one study that showed the promise of using a tumor graft model in personalized medicine. So in this example, a patient with pancreatic cancer had their tumor resected, and bits of that tumor were sliced up and placed into mice. And then these mice were treated with a number of different drugs, both drugs that were commonly used for pancreatic cancer and new drugs that maybe were not normally used. And then the results showed that for this one patient's tumor, some of the mice did not actually respond well to the standard therapy, but they did respond well to a therapy that was less common. And so the patient opted for that less common therapy and then was in remission for over 2 years, which is pretty much unheard of for pancreatic cancer. And so this tumor graft method is extremely promising in terms of guiding therapy decisions for individual patients, as well as for identifying biomarkers and drug targets for cancer. However, personalized tumor grafts are extremely labor and cost intensive. Unfortunately, many patients often die before their mice are actually ready to be treated with these drugs. So it's got a ways to go, but it's promising. So what I've been working on, since mouse models, mice do take time and money, I am working on keeping the prostate tissue alive in vitro in order to do experiments on it, such as with drug targeting and biomarker discovery. And so here's an image of prostate tissue fresh out of the prostate. So this is in its native state. You can see the differentiated prostatic ductal structures with the luminal cells and surrounded by the stromal microenvironment. Now the problem with organotypic culture with the prostate that I've run into is that once we take them out of the body and culture them in vitro, they don't look so good after 5 days. Okay, so depending on different culture conditions, some cells hyperproliferate, like these here, these basal cells are hyperproliferating, and some cells just disintegrate, they die, they don't survive in the culture conditions. And so my goal was to make this look like that. So how do we do this? Well, we harvest cores of malignant and benign tissue from the radical prostatectomies. We place them in this tissue slicer, which generates precision-cut slices about 300 microns thick. That's about the thickness of 3 human hairs stacked. We take these slices and culture them in these dishes here that we place on a platform that is set at a 30-degree angle, and it's constantly rotating. And that allows the tissue slices to move in and out of the culture media so that it's constantly being exposed to the gas, or the oxygen, as well as the nutrients in the media, which contain androgen, as well as growth factors that help keep the cells alive. And once I optimized this culture condition, I was able to keep the both benign and malignant tissue alive up to 5 days in culture. You can see the defined or differentiated luminal and basal epithelial cells, as well as the stroma in the benign tissue. And then, of course, this is the cancer. Now, we verified the differentiation status of these slices using immunofluorescent techniques. Here we have immunofluorescent labeling of the differentiated epithelial cells in the cytokeratin 18, the luminal cells, which are, highly expressed as they should even after 5 days in culture, as well as expression of the normal differentiated basal epithelial cells marked by the p63 staining. And the blue fluorescence is just the DNA. Now, compare the benign tissue to the cancer tissue. We see a decrease in these differentiated cell markers, which just again shows that the cancer tissue of the prostate has been de-differentiated. And this is, however, the cells are they're still alive. You still have the DNA intact and such. So what are we doing with these slices? Well, briefly, One project that we're actually in collaboration with a lab here at UC Berkeley with Carolyn Bertozzi. She's just a fantastic scientist. I hope you cross paths with her at some point in your career. A lot of nodding. And so what we're doing with her lab is using these tissue slice cultures to do glycoproteomic analysis for biomarker discovery using metabolic oligosaccharide engineering. And I have to say, I'm not prepared to describe all this chemistry that the Bertozzi lab does, but you're welcome to, we have a recent publication with them, you're welcome to look that up and keep an eye on her lab to see how that goes. Another project we've been working on with these slices is using them for, down the line, for better drug targeting to cancer cells. We've been doing this by taking libraries of phage that express different oligos or different peptides on the phage and incubate them with normal tissue slices. Then we wash off all of the phage that did not bind to the normal tissue and incubate it with cancer tissue. Then we take the cancer tissue and take whatever phage has been internalized into the cancer tissue, amplify them, repeat, amplify, and that would leave us with a population of phage that have peptides that are specific to the cancer cells. And these peptides we can then take and engineer to express, or to, for example, conjugate them with nanoparticles that contain drugs, or with siRNAs or microRNAs that can target genes, and that way we can target those drugs specifically to cancer cells. If you have any more questions about this or about any of the things that I talked about today, I think we have a question session after the lecture. All right, so to summarize what I've talked about today, altered gene functions or expressions can lead to cancer initiation and progression. These can happen in sporadic or familial mutations. Many of these mutations in oncogenes and tumor suppressors can lead to the uncontrolled progression through the cell cycle or the inhibition of apoptosis. You usually need more than one genetic mutation to cause cancer, according to the multi-hit hypothesis. And transcriptional regulation by hormones can lead to cancer, particularly in breast and prostate cancer. And I also hope I've pointed out that we need better models in order to recreate the genetic and cellular diversity of human tumors. And hopefully using these types of models will lead to better therapeutics, biomarkers, and personalized treatments for cancer. So with that, I would like to thank you for your attention, and thank you to Dr. Ernov.
Thank you very much for a remarkable talk, folks, and those of you who have questions, and I know many of you do. Dr. Mond graciously agreed to stay and answer them in 18010. See you there.